Hello friends, welcome to my tutorial channel once again. I had already given you some questions and answers for ICSE and ISC. This time also I am going to give you some questions and its answers for ISC syllabus. The answers are based on keywords. You have to follow those keywords and answer them accordingly. These are some questions based on inheritance and variation, means the genetics part. The first question is, why are human females rarely become hemophilia? Or why human females rarely become hemophilic? Since hemophilia is a sex-linked recessive trait, this thing you have to remember, sex-linked recessive. If a female has to be hemophilic, the presence of defective genes should have to be present always on both her X chromosomes. But this is a very rare occurrence. Since both the X chromosomes of a female cannot be always having the trait for hemophily, hence human female rarely becomes hemophilic. Next question is, why did Thomas Hunt Morgan, prefer to work with Drosophila for his genetic experiments? We all know that he had done many experiments on linkage with Drosophila. This is because of the following reasons. First thing, Drosophila are very fast breeders. That is, their generation time is about 2 weeks, not more than that. Number 2, they can be grown very easily on a simple media. Question number 3, how can you say that? Sex-linked and caused by sex-linked genes. Number 1 is sex-linked because it shows transmission from an unaffected carrier female to the male progeny. That's why it is called sex-linked gene. Number 2, we can show it is sex-linked because here the heterozygous female may transmit the disease to her sons. That's why it is X-linked. Question number 4 is, distinguish between ZZ sex determination and XY sex determination. Here is the difference. In case of ZZ, the females are heterogametic, but in XY, the males are heterogametic. In ZZ type, the females produce 2 types of eggs, and in XY type, the male produces 2 types of sperms. In ZZ type, males are homogametic, and in XY type, females are homogametic. Examples, as you can see, birds and reptiles belong to ZZ, and Drosophila have XY type of inheritance. Next question is, who proposed the double helical structure? This is the double helical structure of DNA. Who proposed the double helical structure of DNA? What are the different types of DNA? The double helical structure was first given by James Watson and Francis Crick along with Wilkins. There are different types of DNA. The structure that was given by Watson and Crick was known as bDNA. It is having right-handed coiling with 10 base pairs per turn. The diameter is about 22 nanometers, that is about 20 angstrom. Next is aDNA, which was discovered by Rosalind Franklin. This is having a right-handed helix with 11 base pairs per turn. Diameter is about 23 angstrom. Next is cDNA. cDNA, C means complementary DNA to mRNA, which is mostly provided by enzyme reverse transcriptase of the retroviruses. This is a single-stranded DNA molecule synthesized by reverse transcriptase enzyme. And another one is zDNA, which is having left-handed coiling having 12 base pairs per turn and diameter 18.4 angstrom. Our next question. How many base substitutions are possible for all genetic codons and sense codons? All genetic codons, since each base of the codon can be replaced with 3 other bases, as we know that. Therefore, a full codon can be replaced with 9 bases. In case of genetic codons, all the 64 triplet codons can be substituted by 64 into 9, as we said that 9. So 64 into 9 means 576 bases. But sense codons are 61 in number, the other 3 are nonsense codons, means stop codons. These 61 codons are the sense codons, and the total base substitution in them is 61 into 9, that is 549. Next question is, mention the application of DNA fingerprinting. The applications are for identification of criminals and forensic science with the help of DNA collected from samples like blood, hair root, semen, skin cells, etc. from the site of crime, and comparison of VNTR, variable number of tandem repeats. Number 2 is verification of immigrants. It is easy to find out whether the hopeful immigrant is a close relative of a permanent resident or not with the help of DNA fingerprinting. Number 3, DNA fingerprinting can also be used to identify or prove the paternity test, since VNTR of a child matches the VNTR of mother as well as that of a father in different locations. Number 8 is, what are introns? Why do introns need to be separated during the formation of mRNA? The introns are the non-coding intervening nucleotide sequences in eukaryotic DNA. It is separated during splicing because to make the mRNA homogeneous, mature, and fully coding to form proteins. Number 9 is, what are SNRNPs? SNRPs, pronounced as SNRPs. Why are they needed? snRNA means small nuclear RNA combines with short nuclear proteins to form snRNP, small nuclear ribonucleoproteins. These are needed to form spliceosomes required for RNA splicing. Number 10 is, what is the basis of DNA profiling? The difference of the number of tandem repeats and position of mini satellites or VNTRs is the basis of DNA profiling. Number 11. Name the elongation factors in prokaryotes. What sources of energy do they use? These are elongation factor TU, means temperature unstable. Another is EFTS, means temperature stable. And the third one is translocase named EFG. They use both ATP and GTP as their energy resources. If you haven't subscribed my channel till now, please do it. And please share this video to as many students as possible so that they can also have the same advantage as you are having. So please, thanks a lot.